Okay. So we're going to go over a couple things here and basically talk about what's going to be happening next week. Typically, what will happen is I will start off with the major topics we're going to do, but before we do anything else, I will often do a cool science thing of the week, which is something typically relatively recent in literature that's has something to do with what we were talking about. In this case, I picked one kind of random one that's not necessarily something current, very current, but it is still a pretty cool thing. But this is the basis for extra credit for lab practicals. So if you decide to pay attention to the first couple of minutes of lab lecture, then it will be covering basically all of this stuff on your lab practical extra credit. So this one, it's about bioluminescent bacteria responding to different media. So one of the things that we grow bacteria on, we call it media. There's a very wide range of different types, but most of the ones we use in this lab are kind of narrow. They're basically just nutritive. They just provide nutrition for you. However, there are some that can be pretty finicky and needs very special conditions. This one artist, he actually does work with these bioluminescent bacteria. He typically relies on Photobacterium phosphorium, which is it's relatively easy to manipulate. It gives a pretty good glow. It's very common. It's not super dangerous or anything. But the problem is that you need to add salty water and about 1% glycerol to the media to make it continue to glow, which is a little bit of a hassle, especially if you have it contained. So he did one where he put them in these little Christmas ornaments and hung them up. But if you don't constantly supplement them with nutrients, they'll just stop glowing. And so it's kind of a hassle to work with them. They also are really picky about the type of media you use. It's not just seawater and 1% glycerol. It's like those and a special mix of Photobacterium broth that's mixed with agar. It's a pain to make. Now, he was like, okay, well, let's look at some other options. And he settled upon two types of species called Photorhabdus. There's Photorhabdus asymbiotica and Photorhabdus luminescens, which are actually really neat bacteria. They perform the best without any additional carbon sources. So he just put it in there with regular old media, nothing supplemented, and he got a nice luminescence. And the general rule of thumb is for luminescence is if you add something to it, it will glow better because luminescence requires extra resources to make. It's something the bacteria typically only do when they have enough resources to warrant adding in that extra thing. It's not necessary for their survival, generally speaking. So he played around with it a bit, and he added in 1% glycerol or 1% glucose in these ones up here, and he actually saw in increasing concentrations, it actually knocked down the luminescence, which was weird. That's not the rule of thumb. That was very bizarre. On top of that, he also noticed that one of them, when he started adding in supplemental stuff, it actually started producing the secondary dye that nobody had noticed before. It was actually pretty neat. And so it was kind of this really confusing thing. So you can see how even just a small change, 1% glucose, 1% glycerol has a massive impact on the way certain bacteria work. So the components of the media that we use are sometimes extremely crucial. Now I'm going to talk a little bit about Photorhabdus because it's actually a really cool bacteria. Where do we get it? Well, we get it from the guts of nematodes, which infect the guts of these caterpillar guys right here. They're moth larvae caterpillars. There's a couple other ones. And it makes them glow slightly. Why does it make them glow slightly, you might ask? Great question. We don't know. It just does. There's like no reason that we can figure out that makes them glow. Now, I have my suspicions. I imagine that it potentially makes them more easy to be predated upon by, bats or birds or something at night, so that they will come and eat them, carry them somewhere else, and then poop out the hosts of the bacteria, so that then they will have new hosts to kind of go into. So it helps kind of spread them around. But I don't know. Nobody knows. They just happen to glow. Now, they actually also have a human element as well. We know from certain battles in World War I, for example, also in the Battle of Shiloh, that there were soldiers who got glowing wounds. Both of those involved soldiers in areas that had kind of marshy, swampy conditions, which is where this type of bacteria likes to grow a lot. And they noticed that these soldiers with these slightly glowing wounds had a better survival rate than the ones that just had wounds that weren't glowing, and so they called it angel's glow. And in reality, what is happening is that there's most likely these Photorhabdus species, and they produce an antimicrobial agent to help kill off the other bacteria in the wound. However, these bacteria are not really meant to infect humans. So your immune system has actually no problem kicking them out, for the most part, assuming you're a natural, normal, healthy person. They will eventually kick out those bacteria. So the photorhabdis get rid of the bacteria that are in your wound, and then your immune system is able to kick out the photorhabdis, and thus you have a better chance of survival. Pretty neat little mechanism here. Other little note of which to point out, Photorhabdis asymbiotica is actually present here in Texas, and it's actually one of the only times we can mark that we've known in history that there is somebody that has that specific infection because we were able to test for it. It was an older gentleman, and we find that a lot of them are people who like to garden because there's a lot of those microbes inhabiting the nematodes, which inhabit the gardens, because that's where the caterpillars are. So he got a wound on his hand, and it became infected when he was working in the garden, and then it started to glow slightly, which tripped a lot of people out. So now you know, if you ever happen to see somebody in your medical field with a glowing wound, it might be this, or radiation, one of the two, definitely. Okay, so there you go. That's cool science thing of the week. Take-home messages: slight media changes can cause luminescence to increase or decrease. It just depends on the bacteria. So normally we talk about our lab stuff after this. I'll typically have a slide covering safety. In this case, we're going to be talking about microscopy, safety stuff for this. Whenever you guys carry a microscope, make sure you guys use it by 2 hands. Make sure you grab the microscope belonging to your seat number. If you don't know what your seat number is, in front of where you guys sit in the lab, there's a little pull-out drawer that's got a bunch of commonly shared things in it. Inside that pull-out drawer is a number, and it's 1 through 24. So whatever number you have in that drawer, that is your number for your microscope. You grab your microscope out, always use 2 hands. We'll show you guys how to carry it in class, but that's an important thing to know. Carefully handle microscope slides. Place slides back in the appropriate slide box when you're finished. Don't leave them on the microscope. Slides are, of course, glass, and glass breaks, and sharp, broken glass cuts you. So don't drop it. Also, they're really hard to replace. Other ones, clean microscope lenses between slides. This isn't so much a safety thing, but it is a good clean and hygiene thing. It also makes it much easier to utilize the microscopes. So make sure you guys clean them appropriately, which I'll talk about in a moment. And of course, this one, which is only used the fine adjustment knob on the 40x and 100x lenses. The coarse adjustment knob will move the stage up and down too much. You can accidentally crack the glass of the slide or the glass of the lens. Slide don't care about as much as the lens, which is like $100, $200, $300 each lens. So let's be careful of that. Don't raise it up too much. So we only use the coarse adjustment knob. We can use it up and down a lot on the 10x and the 4x, and we use the fine adjustment knob on 40x and 100x. Okay, so microscopes. Generally speaking, you guys should know the general parts of the microscope. They're pretty easy. There's no need for me to go over each individual one, but I will point out something that trips up a lot of people. First one right here is this guy, which is the nose piece. Rotating nose piece. On it is the objective lens. Those are 2 different things. So the objective lenses are the parts that stick out. The rotating nose piece is the part that rotates. Pretty simple. Makes sense. And then underneath this guy right here is the stage. And right here, underneath the stage, is the iris annulus diaphragm and some other adjustment things. But there's stuff underneath the stage. So keep that in mind as well. What we will be doing in class is we will be going at 100x. We will be basically moving through the different powers of the microscope, going from 10x all the way up to 100x objective. The 100x objective is 1,000 total magnification, which I'll talk about in a second. We'll be analyzing blood smears on day 1. So we'll give you guys blood smears to look at. You guys don't have to poke your own fingers, so be grateful for that. But you will slide them in there, and you guys will look around and kind of identify different parts of blood. Blood is very easy to see because it is so much bigger than bacteria, because human cells are pretty big. Day 2, we'll be mastering microscopy skills looking at both gram-stained bacteria as well as a wet mount that we will use for the bacteria that we also will be making on day 1. So we'll have kind of a double thing going on there. First things first, whenever you guys sit down to the microscope, one of the first things you want to do is kind of adjust the oculars. The oculars pull in and out, so each person has their own unique interpupillary distance, which is the really fancy term for that. It's basically how far apart your eye holes are. And everybody's eye holes are a little bit different. So when you sit down, you want to kind of adjust that so you get one clear, large image. So if you're seeing two little kind of weird holes when you look through the microscope, try to make it one big, large hole. The other thing that helps with this is kind of getting down how close your eyes have to be to the oculars. They don't have to be like touching the oculars, but they do have to be pretty close, and there's kind of a sweet spot in terms of distance where your eyes are kind of perfectly seeing a very large circle. If you happen to be a little off to the side, up or down, when you're looking through the oculars, you won't see a full, large field of view. Field of view is what this thing is right here, this large circle where you're seeing. So it takes a little bit of practice kind of getting your head stabilized and kind of getting in the habit of locking your head kind of in place. So keep that in mind. It takes a little bit of practice, but once you guys get it down, you guys will be really good at it, especially if you do it a whole bunch. Okay, so how do you focus a microscope? Pretty easy, pretty simple. Where we're going to start is with the 4 or 10x objective. You don't really have to do the 4x objective. You can start at 10, but some people just like that extra practice, so that's fine too. What you do is you will put your sample, your slide, on the stage, clip it into place. You will clip over the rotating nose piece so that you are looking through the 4x objective. And then you guys will use the coarse adjustment knobs, which are the thick knobs right there. So coarse adjustment is the big guy right here. Fine adjustment is the skinny one inside of that one. And you guys will rotate it forwards until you get the stage. Generally what we like to do is bring the stage all the way up to the top and then slowly bring it down until it comes into focus. Because it's going to be really close to the lens to be able to see. So a lot of people will start up at the top. So if you scroll all the way up so that this mechanical stage is at the top, you look through the oculars and then you slowly scroll back towards yourself and it'll lower the stage. And then you'll see it kind of pop into focus. Once it kind of gets into focus, you'll want to use the fine adjustment knobs to dial it in really well on the 4 or 10x. Really important part here though, once you get it in focus on the coarse adjustment knobs, you're not going to touch the coarse adjustment again. If you do, you have a chance of cracking the slide or the lens, so make sure you're careful with that. Now, one of the things with focus, which is hard for people to conceptualize sometimes, is that bacteria are three-dimensional, much like you. And so there is kind of like a plane of focus where the microscope is going through. So there's a lot of areas where it's like it could be in focus, it might not be in focus. You kind of have to find the right middle spot for that. This is a cyanobacterium. It's kind of large guy, so that's why I chose this one. This is in focus in the plane above the cyanobacterium. This is in focus in the middle of the cyanobacteria, and this is in focus past the plane of cyanobacterium. And you can see you can kind of make out everything in all three of these focuses, but this one gives you the best kind of overall image, and that's the kind of spot that you're looking for. So just keep that in mind. There's kind of like a small range where everything's in focus. Generally, when you bring something into focus, though, you'll kind of see it and it'll kind of pop into sharp focus. That's when you know you want to dial it in and kind of figure it out from the coarse adjustment knob. Once you do that, real quick thing. Once you do that, you will click it over to the 40x nose piece. So you just rotate it a little bit and it'll go to the 40x. You look in there again, and it might not be in focus, and that's okay. The reason why is because when you're jumping up, it takes wherever you were last and brings you roughly into focus on the next one. However, if you were a little out of focus, so before, like we were talking about, if you were a little above what you were looking at, and you pop it over to 40x, you might be completely out of focus when you go to 40x. So if you're on the 10x lens and you pop it up to 40 and you see nothing, what you'll do is you'll take the fine adjustment knobs and you'll turn it a quarter turn forwards, quarter turn backwards. Whichever way is better, keep going in that direction. If neither way is better, you can increase it to a half turn or go all the way up to a full turn. And just keep going in that direction. If you still can't get into focus, then it's time to start back over on the 10x and try to find it again, or ask your TA for help and we'll come help you guys figure it out. Once you get into focus on the 40x, you will then rotate it over to the 100x. Now, the 100x lens, same kind of thing, but even larger kind of thing. So it's even smaller range in the 40x to make it nice and perfect. But here's the really important part for this one. First, this one is an oil immersion lens, which I'll explain in a second. But this one's the one that's really, really, really close to the slide, and there's a lot of potential issues you can have with bringing it into focus. The biggest one that people mess up on is they have the slide upside down. So you can bring a slide into focus on the 10x and the 40x, but not the 100x lens if it's upside down, because the thickness of the glass itself prevents you from bringing it into focus. So if you can't bring it into focus no matter what on the 100x lens, you probably have it upside down. The other thing for the 100x lens is oil immersion. So when you click it in to the 100x, before you click it in, you put it between the 40x and the 100x, you'll put a little drop of this stuff called immersion oil. Your TA will show you guys how to do that. But you just put a drop on there and click the 100x lens in place and let it sit. What this does is it helps gather light, so the light goes straight up into the lens. Because when it's so close like that, it tends to want to like spread out, and you're not getting enough light, and it's just not going to look right when you guys look at it. But if you use oil, it helps direct all the light so it doesn't scatter out this way. It makes it all kind of the same direction that the lens will help make it. So it's basically a much better way to look at it. So if you can't bring it into focus, double check you A, have oil on your lens, or on the slide when you click it over, and B, that it's not upside down. Those are the two biggest common issues. Okay. So once you do that, you click it into place, and you should be able to see basically everything on the slide. There is a video on Lab Archives where I show you guys all the steps on how to go through everything on focusing on the microscope and all that, and all the steps in which you'll see, and some pitfalls and things like that. So I definitely recommend watching through that before you guys come in, but TAs will definitely also show you guys. And really, the first day that we do microscopy is the best day to ask for help, because we will be specifically focusing on that microscopy, so make sure you guys keep that in mind. Okay. Once you have a nice bright picture on your microscope, you're going to, of course, have to document it, and you will document it with your phones. Now, some people can get away with taking a picture through the plastic bags. Some people can't because the plastic bag is too cloudy. So if you find that to be the case, we do say that there's a thing you can do where you can bring up the phone out of the paper bag briefly to take a picture while still holding the paper bag, the plastic bag, take a picture, and then put it back in and seal it. So just, keep that in mind. Now, real big issue that a lot of people are running into is these multi-lens cameras. Believe it or not, this is actually a real camera. It's like 16 camera. That's like a real one. Somebody made that. But the multi-lens cameras on a lot of new phones cause problems with this because it'll be trying to take pictures outside of where the lens is, the ocular lens that you look through. The way to get around this is set your cameras to portrait mode and then set it to 1 or 2x zoom, and that tends to make it just one lens. If you happen to run into an issue where that's not working, definitely let me or your TA know, mostly because we're just still trying to figure out the best way to force cameras to go into one lens, and there's so many varieties of camera. This seems to be the method that works best for most people. What you'll do is you'll hold the phone about 1 centimeter away from the oculars, and you kind of have to find a kind of sweet spot to take the picture. If you happen to, same thing with your eyes, drift a little bit to the right or left or the up or down, it doesn't give you a good field of view, so it tends to help if you hold it really still and use the burst function. I will use my hand sometimes on the side of the oculars and hold the camera still so it kind of stays steady. I have very shaky hands, so I tend to do that a lot. The burst function works well. Some people don't like the burst function, but you can get a good photo. It just takes a little bit of practice, so keep that in mind. Okay. Yes, yes, it is real camera. Because I didn't believe it either. I thought that was a fake picture when I first found it on the internet, but no, definitely real camera. Okay, so human blood smears. What are you guys going to be looking at? Well, first thing you're going to be looking at is you want to look for the fun stuff, which is human blood smear white blood cells. They really stand out. The thing that you're going to see the most of are these guys in the background here, which are red blood cells. Those are really easy to see. They look like red blood cells. Real quick note though, some parts of a blood smear have regions that are kind of globby. So if you're not seeing anything that looks like a nice little pink donut, like normal red blood cells, that's not a good spot to be looking at. And also on the end of the blood smear where it gets kind of really thin, it gets shredded up a lot. And so if you're seeing shreds of pink donuts, that's not a good place either. You want to find that Goldilocks zone, usually right in the middle of the smear. Now, the white blood cells, pretty easy to identify. The first one is a neutrophil. You'll see this most commonly. It's a multi-lobate nucleus. Nucleus that tends to stain purple. So it's super easy to identify. If you see a multi-lobate nucleus, kind of looks like dumbbells chained together to me. That's how I think of it. That's an easy one to identify. The next one is monocyte. Monocytes are these bean-shaped nucleuses. They have a very large nucleus and a lot of cytoplasm, little stuff that's not purple, the little pinkish stuff in here. So that's the cytoplasm. So that's monocyte. The one that people tend to confuse this one with is lymphocytes. Lymphocytes have a very large, round nucleus. and only a tiny crescent of cytoplasm. So that's what differentiates them. They're not that bean shaped, and they only have a tiny crescent of cytoplasm, whereas the monocytes have a large amount of cytoplasm. The last two are kind of interesting, which are basophils and eosinophils. Those are much rarer to find in blood. They're harder to find. I think, what is it? Basophils represent something like 0.4% of blood, and eosinophils represent 0.1% of blood. That's not a lot. That's a very small number. It's hard to find, much like Carmen Sandiego. I don't know if that still tracks with young people, but that was something when I was a kid that someone hard to find. Okay, so they are kind of hard to find, but you guys can find them. To differentiate them is easy. They have one similar feature, which is they have bilobate nucleuses. So they got two little lobes on their nucleuses. You can see here bilobate, and over here bilobate. But they've got these gritty granules in them. And basophils have purple granules and the reason why they're called basophils is because they like basic dyes, and that's why they take it up easily, so they call them basophils. Eosinophils like eosin dye, which is this kind of light pink dye. Thus the name eosinophil. Eosin-loving cell, eosinophil. So these ones have gritty pink granules. You'll tend to see these stand out a lot more, so it's really easy to differentiate them. People tend to see these ones really quickly because they're just really bright. The basophils, people tend to gloss over them. But if you can't see one of them, that's fine. You probably see one of those two types. You don't have to find both of them, but just hunt around and try to get a good view of different types of white blood cells that you can see. All right. Now our day 2 thing is we will have available some bacteria to look at. You guys can look at things like Micrococcus luteus, Bacillus subtilis, Serratia marcescens. These you want to go all the way up to 1,000. So with the human blood smear, you don't have to go to the highest magnification because they're very large, but it's a lot easier to see them when you're at a higher magnification for bacteria. You really won't see much until you're up at that 100x lens, where the total magnification with the oculars and the objective lens is 1,000, just because they're so tiny. So just kind of keep that in mind. Okay, so the second thing. So this lab is a little weird because we have kind of 2 labs in one, which is the microscopy, but we also have this thing called basic handling and culture techniques lab. So we're going to talk about this, and they kind of work in conjunction. So what this one is, we've got some safety things. Most important thing, if something breaks or spills, let your TA know right away, and we'll clean it up together. Probably the worst thing you can do in this lab ever isn't spill something or break something. It's having that happen and cleaning up wrong and not telling us. If something does happen, something breaks or something spills, it's a teaching lab, we expect that. If something spills and breaks and we don't clean it up properly, now we've got a contamination issue that kind of spreads class to class. So make sure you guys let us know. Nobody's going to yell at you or get mad. It's a normal thing. We have a way to clean it up. It's fine. Just make sure you let us know. The worst thing you can do is just hide it from us and not tell us, and then we have a huge issue and we don't know it. Okay. Organisms we will use in lab is considered BSL-1. However, we will treat all the organisms as though they are still BSL-2. It's a good habit to get into, especially when we do work with BSL-2, but most everything we'll be working with is BSL-1. So it generally doesn't have the potential to cause harm to humans or cause any infection or disease in humans. Once items are inoculated, they should go in the incubator belonging to your lab section. There's a label on each of the incubators, but your TA will direct you to which one goes for each of your sections. All stock cultures should be disposed of properly in the proper locations. Your TAs will give you instructions specific to which ones you have, so make sure you guys listen to them. Disinfect your working area and instruments prior to using and afterwards. As always, make sure you guys spray down your working area with Cavicide. Also, make sure you guys wash your hands before and at the end of lab. That's another big one as well, so make sure you guys do that. Okay, so basic handling stuff. Day 1, we'll start with the foundational inoculation techniques. So we'll show you guys how to inoculate stuff. We'll have you guys do it, and then you guys will put them in the incubator and grow them. On day 2, we'll actually use a micropipetter. We'll practice using a micropipetter. I'm sure most of you have used one before. We will use them also to make a slide. It's called a wet mount slide with those bacteria and fungi that we use to make our what we inoculated on day 1. So that's pretty fun. Subculturing. So what is it? It's the process of transferring organisms from one media to another following aseptic technique. Pretty easy definition. There are a couple of rules for subculturing. First one is the tools that you use. Whenever you guys use a needle, you want to be careful what media you're going into. because the needle tends to gouge certain types of agar, so you have to be very careful. So you can use a needle to transfer bacteria from a slant or a broth to a deep, or sometimes to a slant if you want a nice thin streak on a slant, but you want to be careful with that. So a good example of this is for streak plates. So you always want to use an inoculating loop on a streak plate because needles will cut into the agar and gouge it, and then you won't get a nice, clean, even streak. So we always use a loop for streak plates. You can use a loop or a needle for a broth because the broth, you can't hurt it. You can't do anything to it. You just put something in there and it's fine. So broths are kind of the easiest ones out of the ones we'll be using. Okay. Tube inoculation technique. Now, I know it says in here, start with flaming loop or needle. But the first thing we're going to do is we're going to label everything. The reason why is because it's actually a very good habit in science, and I will tell you this from personal experience right now. The second you're like, hey, I'm just going to, I remember what this is. I don't need to label it. I don't need to spend 5 seconds writing down what this thing is. I'll just remember. And then you completely forget, and you have a series of tubes with some cryptic labeling that you might or might not have done very well, or smudged because you didn't do a very good job, and then you have a bunch of things that you don't know what they are, you don't know what it means, nobody knows what it means. Always label your stuff carefully. Also on top of this, whenever we label things, never ever label caps. Caps on test tubes, we reuse those. The test tubes themselves are generally disposable. Some of them are not, but the ones we use in the lab are generally disposable, so we autoclave them and throw them away. The caps are not, and once you autoclave caps with writing on it, it stays there forever. So whenever you guys label, you always want to label on the test tube. So what is the general process for tube inoculation? It depends on what you're going into, right? So if we were going into this, which is a broth, should we use a loop or a needle? What do you guys think? Who says loop? Who says needle? Who says it doesn't matter? Okay, it doesn't matter the correct answer, because it can't hurt a broth, right? If we were labeling a slant, we'd probably want to use, we could actually use a loop or a needle. It doesn't matter as much. It depends on what you want to do. If you want to have just a lot of bacteria to use, then you use a loop. If you wanted to look at the growth pattern of a slant, you use a needle. If we're using it deep, then we'd want to use a needle, because the loop would gouge the agar too much. Okay, so what you guys will do is you guys will flame your loop or needle, whichever one you're using, in the back incinerator. That's those little beige tube things that are on your desks. Those things take a while to heat up, just word of warning, so make sure you guys, if you're going to use them, turn them on right as you come in for the lab, because they take about 5 minutes to warm up. You'll stick it in there, and you only have to stick it in there for like a second, like barely even a second. It's like 400 degrees Celsius inside that tube. It's hot enough to melt metal. Key thing, do not take your loop or needle, stick it in there, and leave it. Because what will happen is, it will heat up the metal, and then it will snap off inside of there, and then I will unhappily have to dig it out. And you could potentially crack the cylinders, and those cylinders are expensive, but they're also incredibly obnoxious to replace. Please don't make me replace them. I hate doing that. It's like the least favorite thing. So make sure you guys don't leave them in there. On top of that, whenever you have a loop or a needle, you never want to put a contaminated one on the ground, like on your table. You always want to decontaminate it right away. So in this case, we have a new one that's from the environment. It's not sterile. We stick it in there, we heat it up, and take it out and let it cool down for a second. Now it's sterile. It's ready for us to use. We will then take a broth culture and our stock culture, so whatever the stock culture is in, it could be in a slant, it could be in a broth. And you'll hold it in your non-dominant hand. I'm not left-handed, I'm right-handed, so I'll hold it in my non-dominant left hand, and you want to hold it kind of at an angle like this, so like I'm holding these pencils. Really weird demonstration, I know, but just pretend. And then you'll take your loop in your dominant hand, you just hold it like a pencil. Hopefully you hold a pencil like this and not like a 4-year-old, but I'm not going to judge, but we're going to hold it like this, and then we'll take off the caps from these tubes with our dominant hand on our pinky and ring and middle finger. So you'll take them off and like that, and you'll hold them here in your hands. You're not going to put them on the tables. If you put them on the tables, guess what? You just contaminated those caps and potentially contaminated your working area with bacteria that was in your stock culture. So don't put them down, hold them, and remember which order you took them off on so that you can put them back on the same order. You'll take your loop, you'll stick it into the tube with the bacteria, you will stick it into the blank media, swirl it around, or do whatever you have to do to inoculate it, and then you will recap the tubes, flame your loop, put your tubes away, don't drop them on the ground, because that would be bad, it makes spill everywhere, and then you put your loop down after it's out of the back to the incinerator. Easy, simple, although I went through it kind of quickly, it gets, the more you do it, the easier it gets. You don't really think about it anymore. You're just doing it from rote. NRTA will give you guys a demonstration on how to do it as well. Okay, streak plate technique. This one requires a little bit more finesse. The reason why we use streak plates is we use to separate it from different bacteria that are like in a mixed culture. Most bacteria that we find in the environment are in a mixed culture. Let me make sure I'm not going over on time. We're doing great. Okay, and so when you guys want to separate them out and get isolated colonies, whenever we see an isolated colony, we can say, that's probably just one bacteria, one genus and species grew that one single isolated colony. And so that's why we'll do this. We also tend to do this when we want bacteria in an actively growing state, so we'll streak them out on a streak plate. There are a couple of different ways to do this. So like this is one methodology they use. This is a bacteria, E. coli, with P. glow plasmid, so that's why it's glowing this nice blue color. But this is the method that we find works best for people who are just starting out. So we're going to show you guys how to do this. Key thing here to notice, these hands right here, is they'll use their non-dominant hand to lift the lid up. Only want to lift the lid up a little bit. The reason why is you take the lid all the way off. Now all the bacteria from the environment, or you breathing, or somebody decides to spit at your plate, I don't know, they'll just go in your working plate, which is not good. That's contamination. So you only want to lift it up enough to get the loop in. And you always want to have the loop in your dominant hand going towards the palm of your non-dominant hand. That gives you the easiest way to control and manipulate things. And I'll explain why in just a second. Oops. So you get your streak plate, you'll draw a line down the bottom and across to make this kind of like three-way pattern here. We always label the bottom of plates. And the reason why we label the bottom of plates is because I can't take the bottom of a plate off. The auger is stuck to the bottom of the plate. If I label a lid, I can take the lid off and put it somewhere else, or put it on a new plate. And now I don't know what this is. So we always label the bottom of plates. Very important. So we'll have our plate, we'll lift the lid up. You'll want to keep your palm right here on this edge, and you'll lift up over here. You'll take your loop, in this case, not needle, because you gouge the auger, with the bacteria on it, and you will streak up and down in this nice little pattern right here. And what this is doing is it's spreading the bacteria out and kind of lowering the number. So by the time we get to the very end here, it's about as low as we can get it with this method so far. We will then flame our loop, let it cool down, turn the plate 90 degrees so that your palm will now be right here. You'll lift the lid up on this end, and you'll stick the loop in, and you will drag it across a couple lines here and do the same zigzag pattern. Key thing, we're not adding more bacteria here. You're actually just moving the bacteria from the first quadrant over into the next quadrant, the third quadrant, so that we can streak it back and forth and lower the number of bacteria there. This will reduce the number of bacteria, so we hopefully get an isolated colony. We'll do this one more time. You will flame your loop to decontaminate it, turn it 90 degrees again, so that your hand will be right here on this end. Lift this edge of the loop up, the lid of the plate up, stick the loop in, drag across these lines, and go in a zigzag pattern. The first little area that we do will probably be a fully dense, like continuous streak of bacteria. But the second and the third and fourth quadrants that we make, hopefully, will be low enough so that you get some isolated colonies. You'll probably almost certainly get isolated colonies in the very last quadrant that you guys inoculate. And that's the reason why we do this. We're just trying to lower the number of bacteria so we get an isolated colony all by itself, not touching anything else. Really, really important part here. Aside from not putting more bacteria in each time you do this, you're only putting bacteria in once. You also want to avoid streaking on the very top of these arcs. There's actually a lot more bacteria on top of arcs for whatever reason, so just keep that in mind. The other thing you want to keep in mind is when you're doing this, you never want to push the loop on the auger. You always want to be dragging the loop. This one takes a little bit more concentration, but basically you always want the loop on its edge pulling it, because if you're pulling it, you can't gouge the auger. But if you push it, now it cuts into the auger and messes up your experiment and makes it look bad, among other things. So please be careful with that. Okay, what you guys will do on day one is we will give you guys a TSA slant, a TSA plate, and a nutrient broth. You guys will label it with your organism, the date, your initials, and the section. Now, you can abbreviate this. So you can abbreviate your initials, you can abbreviate what the bacteria are, that's totally fine. You guys will use a Sharpie to label the test tubes and the plate, but you will not label the caps, very good, or the lid of the plate. So don't label the lids or caps. You'll transfer the bacteria. We'll have two bacteria that you guys will use. You'll transfer E. coli into the slants, and you'll transfer Saccharomyces cerevisiae, which is just yeast, baker's yeast, into the broth. Finally, we'll make a streak plate for isolation of Serratia marcescens, which is a nice reddish bacteria, you guys will see. And you guys will put all of your groups stuff together in the same rack. You guys will put it away in the incubator. On day 2, you guys will take them out, and you will practice pipetting. A couple of things, I'm sure you guys already know kind of the basic parts of a pipetter. But we will be doing a skills check on how to do pipetting for your lab practical, so make sure you guys are actually doing it in the correct volume and kind of know how it works. If you're not sure, make sure you guys let your TAs know. The basic parts of a pipetter is the plunger, which does the sucking up and down of liquid, the area where you put the tip on, which is what actually holds the liquid, and this dial here, which adjusts how much volume you're doing, and this guy tells you how much volume you're getting in. Last little part is the tip ejector button, which moves this little metal thing down here and knocks the tip off. We do have tip boxes that we eject the tips into, so make sure you guys use those when you get rid of the tips because they are potentially contaminated, especially if they have bacteria on them, so make sure you keep that in mind. Setting volumes. Volume starts with kind of a weird, unintuitive thing. You guys have ever used a pipette before, I'm sure you guys know this, but so whatever the type of pipette you're using, if it's a P10 or a P20, that tells you the maximum volume you can go up to. And the first number is always that largest number. So in this case, in the 10 spot, we could bring the P20 up to 20 microliters. And the way it works is it starts with the maximum number, which is the 10s in this case, then the 1s, and then the 0.1s, the decimal parts. So we can set this anywhere from 20 down to usually about 2 microliters is kind of the minimum for this guy. But this one in particular, this one will be set to 15 microliters. So 1, 5, and then this is set to 0, so there's 15.0. P200 here, if we set the hundreds to 0, and this one to 7, this will be 75. P1000, it can go up to a maximum of 1,000. Generally, you don't want to do this unless it's absolutely necessary. So usually this top number will be a 0, but then the next number is the 100. So in this case, it's a 5, so that means it's 500. And on this very last number, it does have these little tick marks. So if I move this tick mark over 1, the tick marks are set up, it depends on your pipette, so that's actually a really important one, but our pipettes are set up by 2s. So if you set it up to the first tick mark, that's, in this case, this would be, if you put it right there, it would be 502. That's not super accurate. Like once you're getting down to the 1s on a P1000, it doesn't work as well, but just keep in mind that's what those tick marks mean. So you have to look at, make sure you're aware of what the tick marks are for each individual one. Okay. Day 2, protocol for this. You guys will practice pipetting 3 separate volumes. Once you guys kind of get a good deal on pipetting and make sure your pipetting is done properly, you guys will then take your bacteria and your fungi from day 1. You guys will transfer 25 microliters of the broth of the Saccharomyces cerevisiae and 25 microliters from the E. coli and put it on a slide. On this slide, you guys will also put a single hair. You can take it from your head, your arm, whatever, and plop it on there. That's just a large thing that's really easy to focus on, so that's the reason why we do a hair. And then you guys will also use a toothpick to scrape the inside of your mouth, basically your cheek only. And these toothpicks are pre-sterilized, so just keep that in mind. So as soon as you use them, you want to throw them away in the biohazard bin. Don't keep the toothpicks around. So scrape off that and streak it back and forth into the broth. And this will create a nice little area for you guys to look at different sized cells. These ones tend to be a little bit harder to see, so it helps a lot of people if they knock down the amount of light going through, like reducing the concentration light, or using the iris annulus diaphragm to change it so that it's a lot easier to see the edges. But basically, you'll be able to visualize both the hair, these large blobs, which is where the cheek cells tend to accumulate, these large round circles, which tend to be the Saccharomyces, because they're kind of these weird oval-shaped things. And the last one, which is what I've seen, is these little dots here that you'll see moving around in the liquid. That would be E. coli, and much smaller than eukaryotic cells, even a eukaryotic cell as small as yeast. Okay, And once you bring that into focus, then you guys will take a picture and then dispose of it properly. All right, now that kind of covers everything. Real quickly, does anybody have any questions on anything we've talked about so far? No. Okay, great. Second thing, very important, make sure you guys complete all of your safety stuff before you come to the lab on Monday, because we won't let you guys come to the lab unless all that safety stuff is done. Of which to note is your quiz 1, which is the basic biosafety quiz, and all the forms that are found on lab archives. Lastly, don't forget to take your pre-lab quiz, which is quiz 2, before Monday at 8 a.m., because that's the deadline. So the deadline is Monday at 8 a.m. for everybody. So make sure you guys take it like Sunday. All right, if you guys have any questions, come up and see me, and I will see you guys later. Have a wonderful, safe weekend.